I'm going to be discussing blood groups today and how blood grouping is done in a lab. Now here we have two patients. One is Mr. Green and the second patient is Mr. Jones. Now we don't know their blood group. So in order to determine their blood group, one of the things we need to do is make this blood interact with something that we do know. So we have this synthetic serum, which is, this one is anti-A serum, which means it contains antibodies against the A antigen. This is anti-B serum, which means it contains antibodies against B antigen. And this is anti-RH serum, which means it contains antibodies against the RH antigen, which is antigen D. So these are the known substances. These two are unknown. Now we're going to mix the two together and then determine the blood group. Now first I'm going to take Mr. Green's blood, and here in this palette you can see 3 wells. So I'm going to pour about 3 drops of blood in one, 3 in the other, and 3 in the third. Now I'm going to add the anti-A serum to the first well up here. I'm going to add anti-B serum to the second, and then I'm going to add the anti-RH to the third. I'm going to mix them, and I have to use separate sticks for each one. And I finally mix it with the anti-RH, and then I observe this to see if there is any agglutination. Now if you look up here, all 3 appear clear. So in the first one, in this one, since there's no agglutination, we know we put anti-A serum, so it would only react if there was an A antigen. So since there is no agglutination, that means the A antigen is not present in the blood. So I put a negative. That means there was no agglutination. In the second one, where I put the anti-B serum, again I see that there is no agglutination. So that means there was no B antigen. So I put a dash there too. Now in the third one, you may not be able to see from that distance, but actually on keeping it for a while, there is a slight amount of agglutination that you can see over here. It is not as clear as the other two. So there is agglutination up here. So that means the anti-RH serum, which contained the antibodies to the RH antigen, found the RH antigen, and hence there was agglutination. Now this is how you're going to map this out for the blood groups. Now let's do another one, and we take Mr. Jones' blood, and again do the same thing. We put 2 drops or 3. Keep this away. And we put anti-A serum. Anti-B serum, and anti-RH. Again I mix them. And I'm waiting to see if I see any agglutination. And I'll just wait a few seconds and observe carefully. Again, from a distance you may not be able to see, but with the anti-A, there was no agglutination. With the B, there is a slight amount of agglutination. It appears opaque. I can't see the writing that I've done over here. So there was agglutination with the anti-B serum, which means it found the antigen. And again, there was no agglutination with the anti-RH. Now let's determine the blood groups having done this. Now if there was no agglutination with anti-A serum, that means this did not contain, Mr. Green's blood did not contain antigen A. There was no agglutination with anti-B serum, that means it did not contain B antigen. But there was agglutination with the anti-RH serum, which means it did have the D antigen. So this is positive blood. Since it has no A antigen and no B antigen, it's O blood group. So this is O positive. Remember, blood group O has no antigens. Let's look at the next one. There was no agglutination with anti-A serum, so that means this did not contain A antigen. His cells did not contain A antigen, so there was no A antigen. It agglutinated with the anti-B serum, so that means it did have the B antigen, so B was present. But again, there was no agglutination with the anti-RH serum, so that means it does not have the RH antigen. So this blood group would be B negative, because there is no agglutination with the anti-RH. So this is how you determine blood groups, and this is the activity that you'll be doing in the lab, where you'll be given the serum and you'll be given unknown blood group, and this is the principle behind typing the blood groups.